so the study of organisms, too small to be seen without the naked eye. We have to magnify it, is microbiology. Microorganisms include bacteria, viruses, fungi, protozoa, algae, and helminths, which are worms. All right. Occupation microbiology, okay, medical microbiology. So medical microbiology studies the effects of microorganisms on human beings, which is if you're going to the nursing program, that's what you're going to be doing. All these little microbes have effects on humans. Okay? Remains the most well-known branch of microbiology. Related fields of public health, epidemiology. So you have public health, epidemiology. Okay, monitor and control the spread of disease in communities. We've had a big surge of this with COVID. You know, everybody's monitoring everything. That's all your public health and epidemiology. Okay, institutions in charge of this hat: the U.S. Public Health Service, the CDC, Center for Disease Control. CDC collects information and statistics on diseases from around the United States, publish it in Morbidity and Mortality Weekly Report. That's where all these COVID deaths are coming from the CDC. Now they're responsible for the flu vaccines. I mean, COVID's kind of taking the main stage right now, but they're responsible for a lot of things, a lot of different drugs, a lot of different testing. The flu vaccine, COVID's the flu. Okay, the flu vaccine is important to get. What they did for the flu vaccine was they guess at the 20 strands or strains of flu viruses that are going to affect people. They put that in the vaccine and they give that out. So can you get the flu when you start the vaccine? Yeah, you can. Because they're only picking 20 of the most common. Okay. And then they give that vaccine out. So, I mean, 65,000 people a year die from the flu. That's not counting COVID. Okay? So what's the big thing? So they go that. Can you get the flu from the flu vaccine? No, not a chance. They use what's called I'm drawing blank on the word. It's attenuated virus, okay? Which means the virus has been killed, and they're giving you the antibodies. And remember, the core DNA has been taken out, and all you got is that little coat, which we'll talk about as we go through our microbiology. But they have that coating around the virus, and that's what you make antibodies to. You don't make it to the virus itself, the RNA inside the virus, the DNA inside the virus. You make it to the outer coating, and that's what they inject you with the flu vaccine. So the flu vaccine cannot give you the flu. Now the COVID vaccine is a lot different. That's a whole different ballgame. We'll go through that as we go along. All right. Field occupation microbiology. Okay, parasites. Okay, parasite specialist examines leaf litter for the presence of black leg ticks, carriers of Lyme disease. Okay. Parasites live in the body and they form a relationship with the host. Usually it's a one-way relationship. Sometimes it's a beneficial relationship. Mitochondria in our cells were once considered a parasite. They infected the cells, they were standalone at one time from what it looks like, and they become a parasite. And that's a symbiotic relationship. That's a relationship where it's beneficial to both host and virus, or host and pathogen. Okay. So a parasite lives off of inside the host. Okay. Biotechnology, genetic engineering, that's the big field here. Okay. Industrial microbiology, using microbiology to make money. That's what big corporations do. These branches revolve around the idea that microorganisms can be used to derive a desired product, anywhere from making beer to vaccines, which we've seen the last year, the big push for the vaccines using microorganisms. Biotechnology focus on your natural abilities of microbes. Okay, so it's just exploiting their natural ability. Genetic engineering involves the deliberate alteration in genetic makeup of organisms to create different types of microbes, different effects of the microbes, plants, animals with unique behaviors and physiology. So we're altering the genetic material, their DNA, to make them produce certain products. If we can infect a person with a bacteria or with a parasite that produces insulin, a type 1 diabetic can't produce insulin. The Langerhans cells in the pancreas are destroyed. If we can infect them with a parasite and that bacteria, we can genetically engineer it to produce insulin. We can have an organism that makes insulin in their bodies. Okay? That's what people have been working on. So there is a lot of medical uses for microbes. Industrial microbiology, scaling any cross produce large quantities of our product. Okay? Microbes multiply much faster rate than humans. Their generations are extremely fast. Okay? They can split cells in minutes. So they can produce a lot of materials. Oil spills, the tanker, the Exxon Valdez that came off there. We can use microbes that eat oil. Okay? We can sprinkle microbes on that oil and they can dissolve that and help protect the environment. So there's lots of uses for microbiology, not just in nursing. Okay? But the main one everybody thinks about is medical microbiology because we think about these things that make us sick. Okay, occupations again, technician tests effectiveness microorganisms in the new production of new sources of energy. We use microbes to produce energy. Okay. Help power, get us rid of dependency on fossil fuels or decrease our dependency on fossil fuels. If you have questions, just speak up, interrupt me. I'm fine with that. I have no problem if you have any questions. So if you ever want to interrupt me or have a question, even if it's not related to microbiology, it's fine. You can go off on all kinds of tangents if I need to. Okay. Immunology. The studies of protective substances reacts to invading microbes, other harmful entities. This is your antibodies, your B cells and your T cells. Okay? How we protect ourselves from parasites, from pathogens. How our body can recognize something that's foreign to the body. I mean, how can we pick up something in the blood that doesn't belong there and destroy it and not destroy ourselves in the process? So it has to be specific. We have to bind to certain antibodies. We'll go over those in a different chapter too. So we don't destroy our own blood or our own cells. Okay? Autoimmune diseases, AIDS, is one of the diseases that affects our immune system. So we end up not being able to identify what's foreign and what's natural, and we dissolve our own cells. It includes areas like blood testing, type A, type B, type O. Okay? Different blood types. You can't mix blood types. You can't give a type B person type A blood. Okay, they're going to find it foreign, coagulate it, and block their arteries, and it will be fatal. Vaccinations and allergies. The allergy is a response to a foreign body in our system, an allergic response. Okay. CDC virologist examines culture of influenza virus. Influenza is one of the main viruses in dealing with the flu. Okay. Producing vaccines works requires high level biohazard containment. If you're working with viruses that cause influenza, that cause a disease in humans, you have to work at a high level biohazard containment. Why would that be? Because you don't want any changes you make in that virus getting out. That's what people suspect happened in China with the COVID virus. Okay? They get out of the biohazard containment. Okay? So when you're working with viruses or microbes, especially ones that are harmful to humans, you have to make sure they're contained. And you have to make sure you don't get infected off. You have to wear personal protective equipment. Agriculture microbiology. Okay? Concerns the relationship between microbes and domesticated plants and animals. Okay? Plant specialists focus on plant diseases, soil fertility, nutritional interactions. And we can alter the growth of certain species of plants. We can alter how animals produce certain things. Animal specialists work with infectious diseases and the interactions between animals and microorganisms. Okay. One of the real deadly diseases is mad cow disease. Okay. We want to be able to limit that and understand what's going on to help prevent that in the future. Microbiologists from the U.S. Food and Drug Administration collect soil samples. We take animal pathogens. Okay. Animals, a pathogen in the soil can get into the animals, get into the meats and the things that we eat. Okay. The Food and Drug Administration is responsible for testing all our drugs. Unless there is a state of emergency declared like with the COVID virus, then they don't have to have human trials for the vaccine to be released. Normally it's a couple of years to get any drug through the FDA for approval. Because you have to go through animal studies, human studies, and it has to be well-designed, double-blind studies to prove that this is effective. And it takes a long.
But for the COVID vaccine, they only had to test it on like animals, right? They didn't have to go through all of that because it was an emergency.
Yeah, it was declared a pandemic, so the FDA didn't have to match the same rules as everybody else. It was released before all the trials. Didn't have to go through all the trials. You're correct. They had done animals and released it. Food microbiologists. concerned with the impact of microbes on the food supply. This areas is food spoilage, foodborne diseases, and production. Okay. Bacteria decrease the storage of our foods, how long they can last. If we can modify the bacteria so they don't affect our food supply can last longer. Okay. Where areas in the world that don't have refrigeration, that don't have ways to keep their food, if we can reduce that spoilage, we can increase the number of lives that we can save by having food last longer. We can fight or inhibit these foodborne diseases. U.S. Department of Agriculture systems are tests for the presence of E. coli. E. coli is a nasty bug. It's not fatal, but it makes you really, really sick. Diarrhea, vomiting, the whole bit. E. coli is not a nice infection. And that's usually a fecal infection from fecal matter. We get E. coli contamination in either water supply from animals or things like that. Origins of microorganisms. Bacteria-like organisms that exist on Earth about 3.5 billion years. So they've been around a long time, way before us. Okay, they've been here for almost since Earth was formed. There's two types of bacteria. There's prokaryotes and eukaryotes. Prokaryotes means pre-nucleus. So they do not have a nucleus. They have DNA, but they don't have a nucleus. It's just spread inside the cell. Okay, so you have prokaryotes, which do not have a nucleus. And you have eukaryotes, which have a nucleus. And those are your complex cells. Okay. This is different. That's a timeline. Shows you how long things have been around. Probable origin of the universe was around 14 billion years ago. Origin of the Earth was roughly 4 billion years ago. 3.5 billion years ago, we get prokaryotes first start to appear, no nucleus. Very simple cells or organisms. Then we get eukaryotes about 2 billion years ago. Okay, they became prevalent on the Earth. Okay. Then we have got reptiles start to appear. This is about 500 million years ago, about 500 million. Cockroaches, termites start to appear. Cockroaches have been here almost forever too. Mammals start to appear, and humans start to appear. And we've been on this for a very small time frame compared to everything else. Okay, so with microbiome, there's two line cells, so lineages basically. So prokaryotes, again, with no nucleus, they're microscopic, unicellular organisms. They lack a nuclei, and they lack membrane-bound organelles. Okay. We'll go through the cell a little bit too as we get some development here. If you have biology, membrane-bound organelles, your mitochondria, your Golgi apparatus, your lysosomes, things like that. Okay. So prokaryotes are microscopic, unicellular, no membrane, and no membrane-bound organelles. No nuclei. Okay. Eukaryotes have a nucleus, unicellular, microscopic, and multicellular. Okay. So we build into multicellular organisms. They have a nucleus, and they have membrane-bound organelles inside the cell. So prokaryotes, before a nucleus, eukaryotes with a nucleus. So then we got viruses. Viruses have been very big on the scene ever since COVID. They're acellular, okay? They're not a cell. They're parasitic particles composed of a nucleic acid and a protein. They don't have life-sustaining materials. They have to infect a host to reproduce, okay? When you think of a virus, think of a little protein coat. This is your virus. There's DNA inside the virus, and it has these little legs on here. Okay? And that's a virus. It doesn't have anything else. It's just a little particle. Okay, it's a protein coat and the nucleic acid inside of it. What we recognize as foreign is these little receptors or little identifiers on the protein coat. This is what our antibodies respond to. Okay? Like I said, with the flu viruses, excluding COVID, the flu virus that you normally get, okay, we take the DNA out of it. We kill this DNA or RNA, whatever's inside of it. So we only inject you with the protein coat. The protein coat is what has the receptors on it where our antibodies bind to make antibodies. So when you expose it again with the real virus, we have antibodies built up in our system, so we kill it before it could take hold. That's how the flu vaccine works. Okay? So viruses are not considered alive. They're not cells of any kind. They're this protein coat with a nucleic acid, either RNA or DNA, inside that protein coat. Okay? Then they attach to the cell membranes and they inject this RNA or DNA into the membrane, and they use the membrane, the host cell's machinery to do the replication of their DNA and doing all their whatever they want to produce. So the figure microbial structure, there's basic cell types. Again, prokaryotic means without a nucleus. You think you'll see that on your exam, what a prokaryote is? No nucleus. Okay. You do have chromosomes. Okay. And you do have DNA. Okay. Those are your chromosomes. That's your DNA inside the cell. It's just not confined to a nucleus. It's spread throughout the cell. You do have ribosomes because DNA takes ribosomes to make the proteins to create your cell wall, to create your cell functions, for the cell to be able to have motion, and the cytoskeleton, cell membrane. You have to create all those through your cell for proteins. Okay? So the chromosomes gets translated, they bind to the ribosomes, and they produce your proteins from your amino acids. You have a flagellum, which allows movement around the cell. You have a cell wall and a cell membrane. Eukaryotic cells have a nucleus. They can have a flagellum. They have a cell membrane. They have ribosomes to make protein, and they have mitochondria. Mitochondria to produce energy. Okay? So this organism can function. We can produce ATP, so the organism can undergo respiration, can undergo movement, can undergo food processing. Right. Now you'll notice prokaryotic cells and eukaryotic cells. Okay, prokaryotic cells have a cell wall. Bacteria have a cell wall. Mammal cells, like our cells, human cells, do not have a cell wall. We have a cell membrane. That's one of the things we use as an antibiotic. We produce drugs. If we can produce a drug that will affect the cell wall, it'll affect the bacteria while not affecting a human cell. Penicillin. Penicillin has a beta-lactam ring that basically punches holes in the cell wall. Bacteria leaks out and it dies. Does not affect human cells because we do not have cell walls. That was a premise for penicillin. That's what it does. So this is a much better picture than I draw. Okay, that's your complex virus. That's called a bacteriophage. Okay, it has DNA inside of it, nucleic or RNA. Okay, so it has an envelope surrounding it, a capsid inside of that, and this is the HIV virus. This is what it looks like. And these are your markers on the side. These are the markers that the body can usually find and recognize as foreign. That's an example of what a virus would look like. Different types of microbes. This is Mycobacterium tuberculosis. Okay, it's a rod-shaped cell. And that's what they look like under magnification. A lot of bacteria, a lot of microbes have different shapes, different functions. So those are rod-shaped cells, and this is Mycobacterium tuberculosis. Okay. Fungi, Histoplasma capsulatum, has like a little lollipop. These little lollipops on the end of each of the structures is their reproductive structure, which allows each of these microbes to reproduce. This is what causes Ohio Valley fever. Histoplasma capsulatum. Algae. Algae, swimming pools that don't run their filters, lakes, ponds, all kinds of fun places. Algae, okay, it's a desmid. This is spirogyrophilament. These are spirogyrofilaments. Okay. These are what's called diatoms. These are what's called golden cells. This is the magnification of algae. You can see the green material, which is chlorophyll. We use sunlight to produce energy. This is a herpes simplex virus. There are coccidiovirus, they're round. This is what a single viral particle would look like. And this is what causes the herpetic lesions on your patient's lips or your lips. Okay. It's extremely common. Most kids carry it. Most people carry it. You usually get infected when you're preschool age, when you share things and put them in your mouth with other kids, and that's how it gets transferred. Okay. You'll notice with the herpes virus, there's different forms, but it usually comes back in exactly the same spot. If you get on the lower left corner of your lip, the next time it comes back, it's going to come on the lower left corner of your lip. It lives in your nerve tracts. Okay. If it lives in your nerve tracts, your body cannot find it. Okay. Your nerves are isolated from the rest of your body. Okay. So if it lives in the nerve tract, it's not going to be exposed to your immune system. All right. So that way you can't fight. So that's why you'll have it for life. But it sits there and it can be inactive along that track. When it comes back, it follows that same nerve path and comes out in the same spot. There's 4 things basically that aggravate or cause the herpes to come out. Okay, sunlight, chocolate, peanuts, and stress. So in the desert, we never have stress and sunlight, right? So those 4 things have been determined to bring out the oral form of herpes simplex virus. Sunlight, chocolate, peanuts, and stress.
So it's contagious? Like it's
Oh, definitely.
like you just,
Extremely contagious.
what
if
if
you
somebody
get-
just has one cold sore like on their lip? Is that a herpes?
Yes, that's a form of it. Yes, they could be on the lips or they can be in the mouth, like cold sores inside your mouth. Like I'll get them on the base of my tongue, hurt like crazy. Okay, they last 7 to 10 days and go away. You find a dentist with a laser. Lasers can treat them. They can decrease their time and decrease their sensitivity. With primary, the
So
first.
if-
Go ahead.
Oh, sorry. So if they're not visible on, like on your lips or something, but someone has had them before, like say you shared a drink with them or kissed them or whatever, like will you still get it or?
No, they have to have the active lesion. The virus has to be transferred. If you kiss someone with an
Okay.
open lesion, you will get it. Yes. If someone has a lesion
Okay.
in their mouth, like at the base of their tongue or on the side of the teeth, and they share a lollipop with another kid at school, that other kid will get it. Or put a toy in their mouth, because preschool kids put everything in their mouths. That's just what they do.
Yeah.
And that's when they usually get it. They usually get your primary infection, and that's usually has a fever associated with it. You feel sick. But after that, you don't get the fever or anything else. It just comes out every once in a while. Some cases more than others. Now, certain diseases will bring them out. HIV, AIDS will bring out, you will get tremendous cold sores. I mean, to the point where you can't eat. You can get 10, 15 cold sores in your mouth. Large ones.
my gosh.
Huge ulcers. Yeah. Very painful. So when,
Okay.
when your immune system becomes compromised, these guys come out. And they are not contagious when they're not active. I mean, if the lesion's healed over, there's no virus here, it's not contagious at all, because they live in the nerve tract. Only when the virus is out and active and being produced, then it's infective.
Okay.
Protozoans. Okay, this is Oxytricha trifolics, bearing tufts of cilia. All these cilia in a liquid environment allow this bacteria or this protozoan to move. It has motion. It circulates the water around it, the fluid around it, and it propels itself through its environment. Okay. Helminth, these are ringworms. Okay. Trichinalia spiralis, okay, trichinosis. Okay. Coiled in the muscle, these worms cause trichinosis. Okay. This used to be a big problem with uncooked ham, uncooked pork. That's when pork was eaten raw back before we knew about all this stuff. Trichinosis was very commonly spread through uncooked pork meat. That's why a lot of religions you don't eat pork because people are getting pork and dying from trichinosis. Okay, these roundworms get infected. But if you cook pork thoroughly, you will not transmit the trichinosis.
How else can you get that? Or is pork the only way?
No, roundworm, you can get infected meat, usually it comes with pork. You can get it walking barefoot through areas that are infected with trichinosis. They can bore through your feet. They can bore through your skin.
Okay, good to know. No more walking barefoot. That's gross.
The Trichinella spiroides has to be there to go burn. If it can go pour through your skin.
All right.
Okay, sometimes the roundworms can bore through your skin, not trichinosis, but the roundworm bore through your skin and cause elephantitis. They'll block the lymphatic drainage ducts. Okay? That you get swelling your legs, your arch extremities, things like that. If the roundworms can get in and block the lymphatic ducts, then you don't get drainage, lymph builds up, and you get huge swellings of different body parts, different limbs. So these are to get you an idea of some of the size of things. Here's an atom. Atoms are really, really tiny. Then you go to a molecule, protein molecule, DNA. These are viruses down here. Viruses are all very small. Okay. Then you start getting your different types of bacteria. Okay. The cocci, the rods, spirochetes, protozoans. Then start the big ones, you get algae, roundworms, and then you get the fleas, which can transmit diseases also. Okay. So this gives you kind of a dimension of size of things. Okay. So these are atomic in size, ultramicroscopic, so scanning electron microscopes, my regular microscope, and these are macroscopic. You can see them without. You can see algae and roundworms and fleas without a microscope. Okay. The flow of energy and food through the Earth's ecosystem. Okay, photosynthesis. Photosynthesis is light field conversion of carbon dioxide to organic materials. Okay. You have carbon dioxide plus water. Carbon dioxide, photosynthesis takes carbon dioxide and water and produces glucose, which we use for food source, plus oxygen. Okay. So photosynthesis uses the energy, that's the chlorophyll in plants and algae. Okay. They take the CO2, which we breathe out, combine it with water to produce glucose, which is other food source, and oxygen, which we breathe. Decomposition breaks down dead matter and waste into simple compounds. So the large cellular components that we have, upon death, breaks down into matter, into the simple compound as the ground, the enzymes destroy it and break it down into smaller, simpler compounds. These are microbes, okay, and energy. This is algae, which you can see. Then the green is the chlorophyll. Absorbs the sunlight, uses that energy from the sun to combine carbon dioxide and water to form glucose and oxygen. Okay. And you see the different areas there, the different types of microbes. Again, microbes are in all kinds of environments, all the way from the Arctic to the volcanoes, anywhere from extreme cold to extreme heat. You're going to find microbes that have adapted to that type of environment. Okay, so which of the following does not describe a fungus? Contains a nucleus, has 80 S ribosomes, it's useful in decomposition, is photosynthetic. Fungi do not produce energy. Okay? It is not photosynthetic. They do not have chlorophyll. Fungi usually go in dark areas where there's no sunlight. They do decompose things. It has 80 S ribosomes, and it contains a nucleus. Human use of microorganisms. Biotechnology. Okay? Production of food, drugs, vaccines. We use living organisms to do that. Because there's a lot of bioengineering is coming out. Genetic engineering, manipulating the genes of organisms to make new products. This is a tremendous expanding field right now. Bioremediation, use living organisms to remedy an environmental problem. You can use that to use organisms to get rid of algae in certain areas. that can dissolve it, oil spills, things like that. Environmental issues that we can use biology, we can use our microbes to manipulate things and create a remedy for that problem.
Did you say that algae helps absorb things?
Algae helps with,
Because
what?
you were saying that, like, can you give an example of how living organism, you said it for oil spills, and
Right,
so we would use certain organisms to absorb the oil?
right. We have used that in the past. We can put microorganisms in there that, because microorganisms feed on, they have to have a food supply, right? Oil spills are hydrocarbons. So they can use those, we get bacteria that feed on oil, break it down CO2 water, because we can break down the carbon hydrogen bond. So we can genetically engineer microbes that can feed on the oil spill and produce carbon dioxide gas and water as a byproduct, and they break it down, which are a tremendous advantage of other problems in the environment. Is that what you're asking?
Yes, that makes perfect sense. Thank you.
Okay. Oh, no problem. We'll get some more examples. I can't think of it off the top of my head. I remember the Exxon Valdez in that one, but we'll get some more examples. Okay. So human use of microorganisms. Okay. What can we do with those? Right. We store them, different cylinders, and then we can use those to modify those, genetically engineer them to create whatever we want to use those for. Okay. So majority live a free existence, are relatively harmless and often beneficial. In many cases, they're essential to life. Okay. So most of the microorganisms are harmless to humans. Okay. They're beneficial in a lot of cases, and we need them to do certain functions. Some microorganisms are closely associated with other organisms, like parasites. Parasites have a relationship with the organism they infect. Parasites live in or on a body of another organism, and the organism is called the host. Okay? And it damages the host. Viruses are parasites. They infect the organism, they live inside of it, and they damage the host. Okay? Roundworms, things like that. Those are parasites. They live inside the body, or they inside of the host of another organism. Microbes and infectious diseases, pathogens. By definition, when something's called a pathogen, it is a microbe that does harm. It harms the body. Okay? It's something that is not passive. Nearly 2,000 different microbes cause diseases. Okay. We have 10 billion new infections every year worldwide. We have 13 million deaths from infections per year worldwide from these 2,000 different microbes. Okay. This tells you the type of disease. It's like influenza. This is your common flu. Okay. 3 to 5 million cases of severe illness, 250,000 to 500,000 deaths per year. Okay. Typhoid, 21 million cases, 200,000 deaths. Measles, about 150,000 deaths per year. AIDS, 1.5 million deaths per year, with 2.3 million new cases. Dengue fever, shingles, viral hepatitis A and B, about 1.3 million. Malaria, 600,000. Tuberculosis, only 1.5 million cases are deaths per year. Now this is worldwide. Remember, there's a lot of third countries that don't have exposure, don't have access to a lot of the medications that we have in the United States or in other developed countries. So we're still getting a lot of deaths, a lot of new cases from these microbes. Okay. So top causes of death. Okay. In low-income countries. Okay. Communicable infectious diseases. Nutritional deficiencies. Take almost half in low income. In high income, it's not even a tenth. Again, we have antibiotics. We have doctor visits. We have things we can do to prevent it. We have hygiene, right, in effect. If you look at injuries, injuries are the same, whether it's going to be in a low-income community, low-income country, or a high-income country. Injuries run exactly the same. Chronic diseases. Chronic diseases are much higher in developed countries. Why is that? Because we've developed sugar. We've developed sucrose. We refine all our foods. We eat fast foods. We do a lot of things for convenience. Chronic diseases are caused by, a lot of these are caused by, we have, in this country, we have obesity at an alarming rate. We have type 2 diabetes at an alarming rate, heart disease, and it all ties into our diet. It's all nutritional because we have refined all our foods to include stuff that we shouldn't have. One of the biggest things you'll see on any food is high fructose corn syrup. Okay? We took regular sucrose, which is not normal sugar, and we've made it sweeter and we made it deadlier because it tastes sweeter and we inject more of it into food to make it taste better. It's all about making money in food. But if you look on a can of Coke, where the first ingredient is HFCS, pick up bread in the store, HFCS, and look at anything. That's the one you really want to avoid if you can. It's extremely dangerous. It causes.
I really try to avoid that.
What's that?
I said I really try to avoid high fructose corn syrup. It's so sad how much they put that in children's products. I mean, everything. It's so hard to
And
avoid
it's a
it.
poison. I was just
You
listening to
literally just have to, you literally just have to eat stuff that comes from the ground, like everything else has it.
Right. What the things you need to do, I mean, I'm not trying to lecture on how to eat, but non-processed foods, no sugar, no sucrose, no high fructose corn syrup, okay? And stuff that is fresh is basically what you should do because all this stuff has this stuff added to it. And it's causing all kinds of chronic disease and it's in high income countries. And that's what our biggest cause of death is. And I was just watching a lecture today out of the University of San Francisco, where he's talking about the effects of sugar and high fructose corn syrup and the effects it has on type 2 diabetes. And that leads to obesity, it leads to heart disease, and arteriosclerosis. It's all of our chronic diseases. And most of it comes from this stuff right here. And like you said, we're feeding that to our kids every day. It's insane. And they know it's toxic. And they just do it because it tastes good. It's sweet.
And it's very addicting, you know. I mean, it's crazy how much these kids crave all of that stuff. And you have to find, yeah.
Sugar's more addicting than cocaine.
They keep making it because they know people
Yep.
will not stop buying it.
That's right. If it tastes good, people are going to buy it. You know, it's crazy. There's a whole series of articles, whole bunch of, everybody's got everything backwards with nutrition in this country. It's all convenient. That's the wrong way to go. All right, so the burden of infectious diseases. So the graph of the burden of infectious diseases continues on the next slide. Chronic diseases include cardiovascular disease, which we said comes from your diet. Cancers, which also comes from sugar, that's a feeding for it. Chronic respiratory disorders, diabetes, neuropsychiatric, sense organ disorders, multi-skeleton disorders, digestive diseases, genital diseases, congenital abnormalities, and skin diseases. Okay, they're not with a single infectious unit, but they're multiple aids that affect it. But if you're not putting the sugar in your body, this high fructose corn syrup, you're not giving a source for these things to take effect on you. Okay. I mean, it's not a cure for everything, but it's a major problem that we have in this country and most developed countries. Okay. So ascariasis we have over 1 billion 1 billion cases. These are neglected tropical diseases. Hookworm infections, which you talked about. 700 million. River blindness, lymphatic fluorosis, sclerosis, trichomia, trichinosis. All you can see all the different numbers that we have for all these different cases. Okay. So we've had thousands of microbiologists over 300 years. There's been a lot of discoveries made. Okay. We do discoveries with the invention of the microscope, the scientific method, how we look at things to make sure we're following a logical path in identifying a problem. Okay. Development of medical microbiology, treating microbes, and microbiology techniques. Those are the things we have developed to help us with our understanding of microbiology and the different microbes that exist around us. Spontaneous generation. Spontaneous generation is an early belief that some form of life could arise from a vital force present in non-living or decomposed matter. Okay. So something that is not alive can spontaneously generate into something that is living. Okay. That's because you had a pile of manure and flies came from it. They didn't realize that flies were there before, they planted their eggs, the eggs would open the maggots go into worms, the worms go into flies. Okay, so the flies didn't come from manure, the flies came from the eggs that were deposited in the manure, allowed to mature there. So spontaneous generation does not occur. We do not get life from non-living decomposing matter. Louis Pasteur eventually disproved spontaneous generation. He proved the theory of biogenesis. Okay, living things can only arise from other living things. Okay. So we do not have spontaneous generation. Okay, this is one of the first microscopes they used. Leeuwenhoek's works. Okay, you had a handle that you held onto there. You had a focus screw which you would twist. Okay, you have a specimen holder and then a lens. The focus screw you would twist and put the different distance between this nail that would hold it in, allow it to move, and allow you to vary the distance between the lens and your specimen to bring it into focus. That was the first microscope, basically. We've come a long way since then. Okay. And you can see different structures. You can see spirochetes, you can see cocci, you can see rods, different types of bacteria, and different types of microbes. Okay. We've also learned to support and use the scientific method whenever we talk about things. We can't just say COVID kills people. We have to show that it does, show what it does, how it does it. Okay? It's been supposed by science to explain a certain natural phenomenon. You form what's called a hypothesis first. You make a guess at something, and then you test it. So you make an explanation that can be supported or refuted. You say, if you get this disease, then this will happen. If you're exposed to influenza, then you will get flu symptoms. Okay, it's an if and then approach. Then you do experimentation, you do analysis of that, you do testing. And the testing results, if it supports it, then you go back and do more and do more. And they continue to support the hypothesis, then it becomes a theory, then it becomes fact. If one of the experiments you do doesn't support it, then it's not going to work. Back to the drawing board and form a new hypothesis. Okay, so results may be published and repeated by other investigators. So it has to be repeatable. Okay, has to be a double-blind study, whereas no one knows what they're having, so it can't be prejudiced. Okay, and if you do it multiple times, when other people do it multiple times, you get the same result. The evidence of a theory is so compelling, the next level of confidence becomes a law or a principle. Okay, so if a hypothesis is supported by a growing body of evidence, you keep doing it in different people, get the results that prove if this happens, then this will happen. Okay, it moves to the next level of confidence, and then it becomes a theory. So we take our hypothesis, and after testing, analyzing all that, if everything keeps testing and nothing proves it wrong, it becomes a theory. So, concept check. A scientific theory has little or no evidence to support it and could be best described as a best guess, true or false. What would you say?
True.
That's how it's a scientific theory. has little or no evidence. A hypothesis has little evidence. A theory has a lot of evidence. Okay? A hypothesis is the best guess. A theory has been proven over experimentation. By experimentation. So that would be false. Right? A hypothesis has little or no evidence to support it and described as best guess. A theory has a lot of evidence that's been tested over and over, and multiple people have got the same result. So that should be false. Vaccinations. How do vaccines work? We observe things, okay, and we make assumptions, make hypotheses, then it becomes a theory. Dr. Jenner observed that cows had a form of chicken pox similar to smallpox. Okay. Jenner noted that milkmaids acquired cowpox only on the hand, and they appeared immune to smallpox. Smallpox was a very fatal disease. Okay. But he noted that people that milked the cows got infections on their hands called cowpox, but they were immune to smallpox. Okay. Scientific method, vaccination, okay. We had the formation of a hypothesis. So he saw this and he hypothesized. He made a guess. He said Jenner deduced that cowpox was closely related to smallpox and could be used on patients to provide protection similar to the milkmaids he had seen. So now you test your hypothesis by experimentation. He took scrapings from the cowpox blister, which is the active form of it, on the hand of a milkmaid and inoculated them into a boy who had not had smallpox. He injected it in. He developed minor symptoms from the cowpox, but he remained healthy. He did not get smallpox. Because they were so close in structure, his body developed antibodies to the cowpox. The antibodies from the cowpox also protected him against the smallpox when it was introduced into the environment. Okay. Experiment 2. After a few weeks, the child was exposed twice to the pus form of an active smallpox lesion. He did not acquire smallpox and appeared to have immune protection. Now, the FDA would never approve this experiment. You can't expose a human being to an infectious disease that's potentially fatal because you want to prove something. But the FDA didn't exist back then, so they could do it. Can't do that today. Okay. That's our first evidence of a vaccine. We got the body to have antibodies to certain proteins on that coat. When he was exposed to it, they're already there, and they killed the smallpox vaccine, smallpox exposure virus before it could become active. Okay. He went on to inoculate 23 other test subjects with the cowpox. For the first time, he used lesions from one child to inoculate another. All subjects remain protected from smallpox. Okay. So this is a graph here. There's a number of cases reporting a number of countries reporting cases of smallpox. In the 1950s, we had 80. 1980s, we had roughly 0. Smallpox is one of the diseases that have been eradicated from the face of the earth. It does not exist anymore. We got rid of the virus by inoculating everybody. Okay. He wrote a paper detailing his experiments. He called his technique vaccination. That's where we get the word vaccine from. It's vaca for cow. Okay. Because it came from the cowpox that he used. Okay. Vaccination became widespread over the next 100 years. Vaccination was brought to the rest of the world through local programs. Scientists use this method to develop vaccines for other pathogens. Now they saw it work for smallpox. They use it for other ones. That's where flu vaccines come from. The theory of artificial immunity became well established. So we give ourselves immunity to different pathogens. As of today, smallpox is eradicated from the world. We use the vaccination, brought it to all the countries, and we have eliminated the smallpox vaccine. The massive vaccination campaign aimed to reduce cases and to stamp out the disease completely. Billions of doses given over a decade reduced smallpox to 0. The last case was in 1977. In 1979, disease was declared eradicated. Now, the problem with that is I'm sure somebody somewhere in some lab is keeping a copy or keeping a contaminated vial with the smallpox vaccine. I don't know why they do that, but I'm sure they are. And if that's released, we're not going to have immunity to it. We figure it's been eradicated with any vaccines. All right, spores and sterilization. Okay, if you work in a medical office, dental office, physician's office, anything like that, you'll have a sterilizer. We have to sterilize our instruments we use on different patients. We use spores to test if the sterilizers are working. It has to be a weekly test. John Tyndall and Ferdinand Cohen each demonstrated the presence of heat-resistant forms of some microbes. So our autoclaves work by heat. So now we have to do something with it because there are heat-resistant forms. Cohen returned these forms for heat-resistant bacterial endospores. Okay. Sterility requires the elimination of all life forms, including endospores and viruses. So we can't just use heat sterilizers anymore. We have to use pressure or we have to use chemicals. And once a week, you take this little packet, and inside this little sealed packet, they have endospores that are live. You stick it in there every Monday, run it with your sterilizer, send it off to the lab. They open it up, look at it, make sure those endospores are dead. If you've killed the endospores, which are the hardest things to kill, you've killed everything else. And that has to be tested once a week in all medical labs to make sure all the instruments that we use are sterilized. So the human body is a source of infection. So when you work on the labs, you work on patients, we use what's called an aseptic technique. That way you don't get other people sick, you don't get you sick with someone that's infected. Yeah. So the human body is a huge source of infections. Dr. Oliver Wendell Holmes observed that mothers of home births had fewer infections than who gave birth in hospitals. Hospitals, you gotta remember this is 100 years ago. Okay. Hospitals do not have the same techniques we have now. People who have diseases, infections, that's where they went to hospitals. And that's what was spread around the hospitals. Dr. Egis Semmelweis correlated infections with physicians coming directly from the autopsy room to the maternity ward. Okay. The washing hands, they didn't wear gloves back then. They went from one room to another. And now that we know that, that spread diseases. So Joseph Lister introduced aseptic techniques to reduce microbes in the medical setting and prevent wound infections in the hospital setting. Involved disinfection of hands using chemicals prior to surgery. They didn't have gloves then, and use of heat sterilization. And now we have to have heat and pressure or heat and chemicals to kill these endospores along with it. Many diseases are caused by the growth of microbes in the body and not by sins, bad character, or poverty. The church had all kinds of theories why people were sick. They weren't good people, so they were being punished, et cetera, et cetera. That doesn't cause disease. If bad choices cause disease, we'd all be in really, really big trouble. There is nobody here who's not made a bad choice or sometime or another. Okay. Now, poverty, you have a higher chance of spreading and contacting diseases because you're living in poor conditions. Poverty doesn't cause diseases, but it has a higher chance of getting them. Does that make sense?
Yes.
Okay. We had two major contributors to these theories for disease of germs, basically. Louis Pasteur and Robert Koch. Louis Pasteur was 1822, 1895. He showed microbes called fermentation and spoliation. That's what causes food to spoil. Okay. He disproved spontaneous generation. In other words, microbes, living organisms cannot come from decomposing matter. If it's not alive, you're not going to get life from it. Another living organism has to produce life. Develop pasteurization. Okay. Pasteurization of milk, getting the disease out of milk before it gets shipped to stores. He demonstrated what's called the germ theory of disease. Okay. He proved that if you didn't wash your hands between patients, you could take germs of one patient and spread them to another, the next patient you saw, or spread them to yourself, or spread them to your family when you came home. Okay. That was Louis Pasteur. Taxonomy. Taxonomy is the different phylization grouping of how things are organized. Okay, so taxonomy organizes, classifies, naming of all living things. Okay, the formal system was originated in 1701 to 1778 by Carl von Linné. It's concerned with classification, orderly arrangement of organisms into groups. And then you've got nomenclature. You have to assign names to these things. Like with tuberculosis. And then you have identification, determining and recording traits of the organism for placement into taxonomic scheme. Organisms placed in similar taxonomic schemes will behave similarly, but cause similar infections. They'll infect similar types of species or hosts. These are the different levels of classification. Your first level is your domain. There's 3 domains. There's archaea, bacteria, and eukarya. Archaea and bacteria are prokaryotes. Okay. There are prokaryotes do not have a nucleus, eukaryotes do have a nucleus. After you separate it in domains, then you have a kingdom, like animal kingdom or plant kingdom. Then you have a phylum or division, then a class, an order, a family, and a genus and species. These are the 2 we use to identify bacteria. We get in the name of a genus and a species. So there's 2 words when we're dealing with bacteria. You can see how the domains are separated. And you can see as they have similar properties, it narrows and narrows and narrows it down. So it comes out to like with humans, Homo sapiens. Homo is the genus, species is sapien. Okay, so organisms in the same family must also be in the same class. So organisms in the same family. So where's our families here? Here's our same family. So organisms that are in this here, that are in the same family, must also be in the same class. Right here. Where class go? Over here. Okay. So if the organism is in the same family, then it have to be the same class. Yeah, we just took that class and narrowed it down. So if 2 species are in the same family, they have to also be in the same class. That would be true. When you do scientific name, they're binomial nomenclature, which means there's 2 names. Like I said, it's a genus and the species. Okay? The genus is always capitalized and the species is all lowercase. Not the whole word, sorry. The genus is capitalized the first letter of the genus. Then the rest is lowercase. Then the entire word of the species is lowercase. Okay, and usually both are italicized or underlined. So Staphylococcus aureus. This is the genus, Staphylococcus aureus is the species. It's 2 words. A lot of times you just agree with just the capital letter S aureus with Staphylococcus aureus. Okay. So it gives you the genus and the species. So it's 2 names that are doing bacteria. Okay. Phylogeny is a natural relatedness between groups of organisms. How close related is one group of organisms to another? How close are humans and the great apes? How close are goldfish and sharks? How close are birds and dinosaurs? Okay. This is how they're related by similarity of the groups. So all new species originate from pre-existing species. Okay. So if we have a new species we identified, it had to come from another species. It didn't grow by a spontaneous generation. Okay. So if you have a new species, there was a species that modified, okay, altered itself to adapt to its environment better, evolved into a new species. So all new species come from pre-existing species. Closely related organisms, okay, have similar features because they evolved from a common ancestral form. Evolution progresses towards a greater complexity, get more and more complex as we evolve and change. so we can adapt to our environment in multiple ways. Okay, traditional organism classification, you got the plant kingdom, plants, fungi, animals. Okay. And you can see where the different genuses and species branch off from the different development. You can see how the branch started down together, and then everything took different divisions as things change. So 3 domains of life: bacteria, archaea, and eukarya. The eukarya have a nucleus and organelles. Archaea are really odd bacteria. They live in extreme conditions. Ice salt, in a volcano, in a geyser, like the geysers in Yellowstone Park, we have bacteria that live in them. The bacteria that have adapted to extreme environmental conditions, extreme heat, extreme cold, Antarctica. Okay, ice salt content. So your bacteria that live in extreme conditions. And the other one is bacteria, which is just your true bacteria. So bacteria, archaea, and eukarya are your 3 domains of life. Evolutionary relationship, you can see the domain. You can see the domain bacteria. You can see all the similarities. Domain archaea, those are your algae, and domain eukaryote, that's what your eukaryotes. Okay, organisms, the domain archaea, have more DNA sequences similar to E. coli, which is domain bacteria, humans, which are in domain eukarya, or archaea, which have no DNA sequence similar to any other organism. Archaea have no DNA sequence from any other organism. Okay. I read the question wrong. Organisms in the domain archaea have more DNA sequences similar to E. coli, which is in the domain bacteria. Which of the following is not considered a microorganism? A, B, C, or D? What would you guess? Archaea,
Mushroom.
bacterium, protozoan, or mushroom?
Mushroom?
Good answer. I would guess that too. Perfect. An area of microbiology that is concerned with the occurrence of disease in human populations is called immunology, parasitology, epidemiology, or bioremediation.
Epidemiology?
Epidemiology is C. Yep. Which process involves deliberate alteration of organism genetic material? Bioremediation, biotechnology, decompositions, recombinant DNA. I would say D, recombinant DNA in the altered genetic material. A prominent difference between prokaryotes and eukaryotes. Remember the difference. So what's it in between prokaryotes and eukaryotes? Larger size of prokaryotes, lack of pigmentation in eukaryotes, presence of a nucleus in eukaryotes, or presence of a cell wall in prokaryotes. A prominent difference between prokaryote and eukaryote cells is the
Presence of a cell wall?
say presence of the nucleus. That's the main difference. But there is a cell wall also. But some prokaryotes still have cell walls, some bacteria. So I'd say the presence of a nucleus in eukaryotes. Prokaryotes have no nucleus, eukaryotes have a nucleus. Which of the following parts was absent from Lekker's microscope? The focusing screw, a lens, specimen holder, or a condenser. Remember the little screws you held up, but when you twist the little screw and with your little lens, the first microscope, what was missing? You had a focusing screw, you had a lens, you had a specimen holder, you had no condenser. It would be D. A, biogenesis refers to the spontaneous generation of organisms from non-living matter, development of life forms from pre-existing life forms, development of aseptic technique, germ theory of disease, sorry. Which one is A, biogenesis?
Spontaneous generation of organisms?
From non-living matter. You got it, A. Hypothesis, where am I at? Hypothesis can be defined as a belief based on knowledge, knowledge based on belief, a scientific explanation that is subject to testing, a theory that has been thoroughly tested. So what is a hypothesis?
C.
C, a scientific that is subject to testing. Yep. Which early microbiologist was most responsible for developing standard microbiology laboratory techniques?
C.
I don't remember names. Let's try C. It's B, Robert Koch. Which scientist was responsible for finally laying the theory of spontaneous regeneration to rest? Who finally destroyed it?
Was it D?
Pasteur or Lister? D or A? Remember which one? D, Louis Pasteur. Very good. Perfect. Louis Pasteur. When a hypothesis has been thoroughly supported by long-term study and data, it's considered a law, speculation, theory, or proved. What would it be?
Theory.
It'd be a theory, C. Which is the correct order of taxonomy categories going from most specific to most general? You could just look that up anytime you want, but it looks like domain, kingdom, it looks like A to me. Let's see. Oh, going backwards. I was going the other way, sorry. Okay, so going from most specific to most general. Species, genus, family, order, class, phylum, kingdom, domain. I was going the wrong way. It is C. By definition, organisms in the same what are more closely related than organisms in those in organisms in the same
Family and
family
Genesis.
and genus,
Genesis.
class and phylum.
Oh.
Class and phylum. I have to look that back up too. Which of the following are prokaryotic? Bacteria, archaea, protists, both A and B.
Archaea and bacteria.
D.
I don't know if archaea is. D, both, you're right. Archaea and bacteria are both are prokaryotic. You were right. And then this one, you have to list the order of the numbers, and you just have to list the size. The atom is the smallest, and the largest would be protein worm. Largest would be rickettsia. And then you just look up the size of the prokaryote and determine which one's bigger than it. And which defines not an emerging infectious disease? Avian influenza, Lyme disease, common cold, West Nile virus. Which one is not emerging as infectious? It would be the common cold. Everything else is an emerging infection. Yep, common cold. How would you characterize a virus? A prokaryote, a eukaryote, an archaean? None of these.
None of these.
None of these. Yep. D. It's not a cell. It's not alive. It's just a protein coat with an RNA or DNA. A DNA series of nucleic acids inside. A small section of DNA or RNA inside. Okay, let's do the case studies. Most of the bacteria. And then we'll do the test one that should do it. All right, so most bacteria in Lake Woodlands derive energy from oxidation of chemical compounds. So you have bacteria in the lake, they're deriving energy from oxidation of chemical compounds. They're breaking down chemical compounds to get energy. Why would they do that? Because they have exceeding low temperatures, lack of sunlight, the size of the population they found, or lack of fresh water. What would cause bacteria to not use photosynthesis, but use the oxidation of chemical compounds in the lake?
The lack of sunlight.
Lack of sunlight.
Agree. Lack of sunlight. How was the number of different species in the lake determined? I'd have to read the study, which we didn't do, but that's probably in the book. Is by analyzing DNA recovered from the samples. And do you think scientists working at Pitts Lake were at great risk of infection from the organisms growing in the lake? Look at that. That appears in the index and read the thing. All right, so here's one question for your nursing, your NCLEX or your TEAS exam. So the nurse in an emergency room is reviewing the discharge instructions for a client. The client asks for a clarification of zoonosis with regard to the type of illness. What is the best response by the nurse? A zoonosis refers to any viral disease. A zoonosis is usually referred to that a disease that can be successfully treated with antibiotics. Zoonosis is disease typically found in animals that can affect humans. Zoonosis is a disease that causes by eukaryotic parasites.
Is it B?
B or C? I would say C. Yep, C. Zoonosis is a disease typically found in animals that can infect humans.